Gel electrophoresis is an easy way to separate DNA fragments according to size. Firstly, we put a gel and we run a current through it. DNA is negatively charged, so once we place the wells at the negative electrode, what happens is that each well, we put our specific DNA in there together with a dye so that we can see it. We place the gel and we add buffer to the gel box. This is known as a gel box. And then we cover it and switch on the electricity. After about 30 to 45 minutes, we take the gel out and we put the gel into a under UV light, and this is what we see. So the wells were somewhere in this position, and now the DNA has separated. Now, in this particular case, we looked at sea urchin genetics and at a specific gene of interest, and we actually had black colomorphs and red colomorphs at certain location on the reefs in Trinidad, and we can see that there are the same species with respect to this gene. And so, what am I talking about here? If we think of the scientific method, observation is pretty much where we eventually ask the research question. So on the reefs in Trinidad, we see different color of these soft corals. Notice there's a green, there's a brown, orange, and then we use the genetics, the gel electrophoresis, the polymerase chain reaction, and then through DNA sequencing, we send that off to a company, and guess what? Here we can see certain species of soft coral. So here's Palithoa, but brown, green, and brown from different Toku Bay, Sally Bay, Western Sally Bay. And notice they are all the same species after we've looked at its phylogeny. All right, we use an outgroup, another species, to show that it is genetically different.